So, welcome everyone. Good morning, afternoon, or evening, depending on where you're tuning in from. Thank you all so much for joining our IF-IHC workshop today. The focus of today's workshop will be the importance of the proper selection of secondary antibodies for immunostaining. So just some intros. I'm Afrida Rahman Enyart. I'm a product manager at Proteintech, and I will host today's webinar. Our speaker running the workshop today will be Dr. Swati Roy, who is the product manager for our recombinant antibody, secondary, and immunohistochemistry portfolios. And also joining us is Abel Zhang from our R&D department, who will be here to help answer, all your technical questions. So before I pass it off to Swati, to begin the workshop, I do want to share a couple slides introducing Proteintech. So if we could go to the next slide, please. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies encompassing over 14,000 targets. We also offer ELISA kits such as ELISA kits, IHC kits, and also antibody labeling kits. Within our Proteintech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells. And so this is going to be different from when you see proteins that are expressed in E. coli because they're going to be in their human native form. So they have native folding, post-translational modifications, and glycosylation. We also have our Chromatech branded nanobody-based reagents. And nanobodies are about 10 times smaller than traditional antibodies. And because they're so small, they're really great for things like super resolution microscopy. And they also bind to their targets with really high affinity. So they're great for clean immunoprecipitations as well as protein purification. And then lastly, we also have introduced Proteintech Genomics. So Proteintech Genomics is a subsidiary of Proteintech, and they manufacture antibodies that are conjugated to oligonucleotides for single-cell RNA sequencing. If we could go to the next slide. So our motto has always been that we are a company for scientists by scientists. So our high quality and award-winning products are self-manufactured, and we offer the largest in-house portfolio of antibodies for the human proteome. We also know the importance of product validation. So whenever possible, we validate our antibodies using knockout knockdown models. Also, as antibody experts, we have in-depth knowledge of immunoassay applications and can offer outstanding PhD level technical support. And then over to the next slide, I would like to introduce our expert instructors for today. So Dr. Swati Roy and Abel Zhang. So Swati, who will be leading our workshop today, joined Proteintech as a product manager in 2022 and currently manages our recombinant antibody and immunohistochemistry portfolios. Before joining Proteintech, Swati worked at a cell and gene therapy CRO named Vector Builder, where she started off as an application scientist and then later transitioned to a product manager role. Before starting her career in the biotech industry, Swati graduated with a PhD in biology from the University of Houston. We also have Abel Zhang here with us today, who will be assisting with the Q&A. Just a little bit of background on Abel. Abel comes from our R&D leadership team. He has 16 years of experience in antibody development and validation. And his team has developed over 3,000 high-quality monoclonal and recombinant antibodies, as well as top-class recombinant secondaries. So collectively, Swati and Abel can bring a lot of technical knowledge and expertise to the table, and I really hope that you all get to take advantage of that today. So if at any point during the workshop you do have any questions, please feel free to input those into the chat or the Q&A box, and either Abel or Swati will address them via the chat or live at the end of the workshop. So with that, I'll go ahead and I'll pass it off to you, Swati.
Thanks so much, Afrida, and hello, everybody. It's great having you all here today. So I'll go ahead and jump onto today's topic. So pretty much as Afrida mentioned, the focus of today's topic would be the importance of selecting the right secondary antibodies for your applications. So generally speaking, the overall agenda would be just to give you an introduction to secondary antibodies and indirect detection. Next, factors that need to be considered when you are selecting your secondary antibody. And lastly, I'll also be giving you you an overview of Proteintech's multi-RAB recombinant secondary antibodies, which are kind of the most advanced class of secondary antibodies that we have developed and the advantages offered by them. And as Afrida already mentioned, that please feel free to pop in your questions in the chat throughout the session, and or we can take them at the end of the talk. So with that, moving on to the first topic, which is just a general introduction of secondary antibodies and their importance in indirect detection. So as you all are already aware that secondary antibodies do facilitate indirect detection of target molecules by binding to primary antibody, which in turn binds to the target antigen. So you can see the graphic over here. Basically, it just shows you what's the basic difference between direct and indirect detection. In direct detection, you have the primary antibody conjugated to your label, which can be an enzyme or dye, which directly binds to the antigen. Whereas in case of indirect detection, you have your primary antibody, which is unconjugated, binding to your antigen, and this is followed by a secondary antibody step, which in turn is conjugated to your label. And the purpose of having a tag or label is that it typically facilitates detection or purification. So just before jumping into kind of the, factors or considerations of selecting secondary antibody, just to give you a very generic overview of how the indirect detection workflow works for IF and IHC, this can be quite variable, especially in the sample processing step, depending on whether you're working with cells or tissues, or you're looking at an intracellular target, membrane target, things like that. But generally speaking, the steps involve collection of your sample, which can be cells or tissue. Then, of course, you have to fix your sample to maintain the physical and chemical properties of your sample. Then comes the sample processing part, which as I mentioned, can be pretty variable. It can involve a permeabilization step if you're looking at an intracellular target, or if you're doing, let's say, fluorescent IHC, it can involve embedding into OCD media. If you're doing IHC for FFPE samples, then it's a lot more elaborate, where you might have to perform, dehydration steps, paraffin embedding, sectioning, rehydration, and antigen retrieval. So all of that kind of goes into this sample processing part. And this is followed by a blocking step, which prevents any kind of non-specific binding of the primary antibody. This is followed by primary antibody incubation, which in turn is followed by secondary antibody incubation. And then sometimes we also have an amplification step. So I will be talking about the amplification step later on during the presentation, because this does have an impact on what secondary antibody you're selecting and how it can significantly help to amplify your signal. And then finally, you have your counter staining step, where you usually, stain your nuclei with DAPI or hematoxylin, and then you do your mounting. So that's kind of the overview of the process of how the indirect detection workflow works. Now, of course, we put in a lot of thought in selecting our primary antibodies, but there can be a lot of factors which goes into the secondary antibody selection, which can also have quite a bit of impact on your experimental results. So I'll be talking about those today. And I do also want to emphasize here that, as you can see, that the having the secondary antibody step in indirect detection does add an extra step, right? Like by doing direct detection, you can eliminate that step, but there are still like several labs, are in fact a higher number of labs, which still prefer the indirect detection method. And the reason being is that indirect detection does offer quite a few advantages. So first being increased sensitivity. So in indirect detection, what you have is multiple secondary antibodies can bind to a single primary antibody. So at least two secondaries are bound to every single primary antibody, and each of those secondary antibodies are bound to one or two tags or labels. So this results in amplification of signal. Indirect detection is also much more versatile, which means that you can use the same secondary antibody to detect multiple targets by pairing with different primary antibodies in different applications. Now, as an example, you can see here that we use our multi-Rab goat anti-mouse recombinant secondary antibody for detecting a lot of three different targets across three different applications. So the first one is just immunocytochemistry, second one is fluorescent immunohistochemistry, and the third one is flow cytometry. So the same secondary antibody was paired with different primaries across three different applications. Now, one thing I just want to mention here is that a lot of data that I'll be showing you throughout the presentation today will be with these multi-Rab secondaries, and I'll talk about them, what they are, and how we develop them at the end. But just keep in mind that in terms of from a protocol perspective, it is no different. So you would use a multi-Rab recombinant secondary antibody in your IF or IHC workflows exactly same as you would do with your polyclonal secondary antibodies that you typically use. So next advantage of indirect detection is that you also have more flexibility. So you can pair the same primary antibody with various secondary antibodies depending on the intended application. So you're not just stuck with one application. You can have the same primary. So in this case, in this example here, we use our TDP43 N-terminal recombinant primary antibodies in combination with various secondary antibodies for three different applications. So for example, HRP conjugated secondary was used for Western blot in the first case. A poly-HRP conjugated secondary was used for immunohistochemistry, and a correlite conjugated secondary antibody was used in the third case for fluorescent immunohistochemistry. If you're not aware of the correlite dyes, just I want to mention here that correlite dyes are our proprietary dyes that were developed in-house by Proteintech. So, and they spectrally are very similar to Alexa flow dyes, which are more, you might be familiar with already. And lastly, the last advantage is the easy availability, right? So when you're working with direct detection, you will require a primary antibody which is directly labeled with your conjugate of choice. So that can sometimes be limited. You might have limited options, but on the other hand, for secondary antibodies, we have a lot more app options. Secondary antibodies against most species are widely available in a variety of conjugate options from multiple commercial suppliers. So Proteintech, for example, ourselves, we offer 10 different species reactivities, 5 host species, and more than 15 conjugates, which includes our correlite dyes, a lot of the traditional dyes like FITC, APC, PE, and then also enzymes like HRP, poly-HRP, or alkaline phosphatase. So you have a lot more options to work with when you're working with indirect detection. So with that in mind, so that kind of sets the background for what the importance of secondary antibodies is in indirect detection workflows. Now, moving on to the most important part of today's webinar, that what are the factors that you need to take into consideration when you are selecting your secondary antibody? So in coming to factors, I will be talking about 6 different topics today. So the first being target specificity, which refers to species, class, and subclass specificity. Second will be host species, so species in which the secondary antibody was raised. Third is antibody format, so whether you're going to use a whole versus a fragment antibody. Fourth is clonality, whether you want to use a polyclonal or a monoclonal secondary antibody. Conjugate, which can have a lot of impact and varies by application, target, and also the properties of the conjugate itself. And then cross-adsorbed. So what are cross-adsorbed secondaries, when you should use them, and are there any alternatives for cross-adsorbed secondaries? So I'll be going through each of those points one at a time, and if you have any questions, feel free to just pop them in the chat. So target specificity, so very simply speaking, when we talk about species specificity, it just means that the secondary antibody must be raised against the host species of the primary antibody. So just as a very simple example, in this data over here, you can see that it was stained with two different primary antibodies, rabbit anti-alpha tubulin and mouse anti-NPM1. So the two hosts of the primary antibodies were rabbit and mouse. So we used our multi-rab anti-rabbit and multi-rab anti-mouse recombinant secondary antibodies for the detection. So if you are having two different host species, you have to make sure that your secondary antibodies are raised against the host species of your primary antibody. So that is pretty straightforward and simple. Now, what about like when should you take class and subclass specificity into consideration? So generally speaking, yes, the primary antibody, the secondary antibody should be specific for your primary antibody class, which is also known as isotype or subclass. So let's say when we talk about rabbit polyclonal antibodies, they are typically of IgG isotype. So generally speaking, rabbits have four different isotypes or classes of immunoglobulins, which include A, E, G, and M, but the polyclonals are usually of IgG origin. And for rabbits, luckily, they have just one single IgG subclass. So the commercially available anti-rabbit secondary antibodies can very efficiently detect the IgG isotype. So you are good with that. But when it comes to mouse, it's a little bit more complex because mouse not only has five um, isotypes or classes of immunoglobulins, which is like IgG A, IgD, IgE, IgM, and IgG, but the IgG isotype also has five different subclasses. So most of the mouse monoclonals are of IgG isotype origin, and these can have five different subclasses, right? IgG1, 2A, 2B, 2C, and IgG3. However, the good news is that regardless of whatever IgG isotype your primary antibody is, most commercially available anti-mouse secondaries can recognize all Ig classes and subclasses. So as an example, I have shown here that we have used our poly-HRP conjugated anti-mouse recombinant secondary antibody for detecting four different mouse monoclonal antibodies. And as you can see that each of these four antibodies have different IgG classes of subclasses. So the mu N, for example, is IgG1 subclass. Then we have a IgG2A SLC22A7 antibody. And then we have a IgG2B CD107 or LAMP1 antibody. And the last one, the MPO antibody, is actually of a completely different Ig class or isotype, IgGA. But the same secondary antibody was able to successfully detect all of the IgG subclasses and classes from mouse. So so generally speaking, that is just to say that regardless of what origin your primary antibody is, the anti-mouse secondary antibodies that you're using should be able to recognize your IgG class or subclass. So when do you want to take the IgG subclass into consideration? So that really comes into play if you are trying to do multiplexing between different IgG subclasses. So what these subclass specific secondary antibodies do is they enable multiplexing between different IgG subclasses. So you do have commercially available antibodies available, which are specific for mouse IgG subclasses. Proteintech offers subclass specific nanosecondary reagents. I think Afrida mentioned in the beginning. So nano secondary reagents are from our Chromotech portfolio, and they are derived from recombinant VHHs or nanobodies, which are single antigen binding domains from camelid-derived heavy chain only antibodies. So we do have subclass specific nanosecondary reagents, and these are available against mouse IgG subclasses IgG1, 2A, 2B, and 3. So if you look at this dot blot or immunoblot on the left here, you can clearly see that each of these subclass specific nanosecondaries show no cross-reactivity for other mouse IgGs. Similarly, for in this fourth row here, we also have our anti-rabbit IgG nanosecondary, which also doesn't cross-react with any of the mouse IgG subclasses. However, if you look at this last row over here on the immunoblot, you can see that it shows a pre-adsorbed goat anti-rabbit IgG from a competitor. So pre-adsorbed antibodies are generally those that have been subjected to an extra purification step to eliminate any cross-reacting species of antibodies. So even though this secondary antibody at the last was pre-adsorbed against mouse IgGs, it still shows some minimal levels of cross-reactivity with different mouse IgG subclasses. So that is to say that our nanosecondary reagents are very suitable for multiplexing between same species antibodies. And as an example, you can see here that we have HeLa cells that are co-stained with two mouse antibodies. One was IgG1 subclass, the other was IgG2b subclass. And we used IgG1 specific and IgG2 specific nanosecondary reagents for detecting them. So moving on to the next factor. So that was a specificity of for your species, class, and subclass. The next factor that has to be considered when selecting your secondary antibody is your host species. So very, very important is that your secondary antibody host must be different from the primary antibody host. So typically you will see that when your primary antibody is raised in mouse or rabbit, your secondary antibody is usually originated from either goat or in donkey. So typically, secondary antibodies raised in goat or donkey are most widely used because they are suitable for the detection of primary antibodies from most species. Now, that's pretty straightforward, but just one thing to keep in mind is that when you're doing multiplex experiments, you have to make sure or at least try to use the secondary antibodies that have originated from the same host, because this helps to avoid cross-reactivity with other secondary antibodies. So what I mean by that is if you look at this data figure over here, here we used a rabbit anti-New-N antibody and a mouse anti-Calbindin D28K antibody. But the secondary antibodies that were used, of course, we used anti-rabbit and anti-mouse multi-Rabs, but both of them were raised in goat. So that's very, very important. So you have to make sure that if you are doing double or multiple labeling experiments, your secondary antibody originates from the same host species, because that can significantly avoid cross-reactivity between the different secondary antibodies. Next, moving on to the third factor that needs to be considered is antibody format. So what does antibody format means? Antibody format, as you can see from the illustration over here, that on the left, you see how a whole immunoglobulin molecule or antibody molecule looks like. Now you can subject your antibody or whole antibody molecule to different types of proteolytic treatments like pepsin digestion or papping digestion to kind of yield these different fragments. So when you subject them to pepsin digestion, you still have your antigen binding domains connected by the disulfide bonds, which yields a Fab2 fragment. And then when you do a papain digestion, you can actually also cleave off the disulfide bond, and you can have these single or monovalent Fab fragments. So when do you actually or when is it recommended to use these fragment antibodies versus whole antibodies? So for most cases, the whole antibodies are the ones which should suffice, and that is the most commonly used secondary antibody format. But in certain situation or certain applications, it has been seen that a fragment specific primary antibody can actually help to enhance the signal to noise ratio. So if your primary antibody is fragment specific, like let's say your primary antibody is specific for the Fab fragment, fragment, then it might be also or helpful to use a fragment secondary antibody to prevent any kind of background noise. Fragment secondary antibodies are also recommended if you're staining tissues that are rich in Fc receptors, because obviously, as you can see from the structure, these fragment antibodies lack the Fc region. So that helps to eliminate any kind of background by the non-specific interaction of the Fc domain with samples such as lymph node, thymus, blood, which might be rich in Fc receptors. So that's another situation when you might want to consider using fragment antibodies. And the third thing to take into consideration is that fragment antibodies are also much smaller than whole antibodies. So, for example, the Fab fragment is about 1/3 of the size of the whole antibody, and the Fab 2 is 2/3 of the size. So, because of this smaller size, they can penetrate tissues more efficiently, and that's why they are a preferred choice in IHC applications many times. So that's kind of the background on whole and fragment antibodies. Now, very similar to the fragment antibodies here, we have our nanosecondary reagents. So a lot of the advantages that our nanosecondary reagents offer are similar to fragment antibodies. So as I mentioned earlier, nanosecondary reagents are highly specific recombinant alpaca single donor VHHs, right? So they basically consist of single antigen binding domains from heavy chain only antibodies that are derived from alpacas, and they bind with high specificity to Fab or Fc fragments of primary antibodies. You can see the kind of the size comparison of a nanobody with a whole antibody over here. They are about 10 times smaller than conventional IgGs, and because of this smaller size, they offer advantages of minimal label displacement. This means that because of their small size, the distance between the label or the conjugate attached to the secondary antibody is much lesser from the antigen compared to when you're using a full-length primary antibody. And you can see how the complexes look like when you're using a primary antibody nanosecondary combination versus when you're using a primary antibody and traditional polyclonal secondary antibody combination. So because of the small size, it does offer several advantages. So the first is just like the Fab or Fab2 fragment antibodies, because of the smaller size, nanosecondary reagents also enable better tissue penetration. And this is very helpful when you're working with tissue samples or organ samples or whole organism samples. So this is an example of data generated by one of our customers showing staining in Drosophila larval brains. And on the left, you can see that it shows a combination of anti-lamin primary antibody with nanosecondary reagent, whereas on the right, you see the same primary antibody was combined with a traditional anti-mouse secondary antibody. And you can clearly see that there is a difference in the coverage or the staining pattern, or you can see much higher coverage when the nano secondaries were used compared to traditional secondaries. Next, because of the smaller size, as I mentioned, that nanosecondary reagents can achieve higher, like they have minimal label displacement. This also allows them to achieve higher resolution. So that's the reason that we highly recommend using these nanobodies or nanosecondary reagents for any kind of super resolution imaging application. So this is an example where it shows comparison between confocal and STED imaging of nuclei and HeLa cells. And you can clearly see that the resolution achieved with STED is much higher than in confocal when you use the nanosecondary reagents. So again, that being said, that these are highly recommended for super resolution imaging. And this example over here also illustrates the same thing. This was also a customer generated image. It's a 3D sim image of photocytes, which again shows that the resolution was pretty high when they used nanosecondary reagents. And the customer also commented that they were able to increase resolution by about 20 nanometers parameters with very minor background when they use nanosecondary reagents. So just to conclude about this point, that you might want to take antibody format into consideration when you're working with specialized applications. And for super resolution imaging applications, the nanosecondaries are definitely a very good option. Next, moving on to the next factor is clonality, right? So clonality, meaning whether the antibody is polyclonal or monoclonal. Generally speaking, we don't think about clonality quite a bit. Like when it comes to the choice of secondary antibodies, we think about that more in terms of the primary antibodies, but it can impact your data quality in certain situations. So just before we get into that, just to kind of refresh your memories, what are the differences between polyclonal and monoclonal antibodies, and also nowadays recombinant antibodies are very prevalently being used, are being preferred over both these two traditional classes of antibodies. So what is the difference between these different classes of antibodies? So in terms of source, polyclonal antibodies are obtained from the serum of an immunized host animal, for example, rabbit. In terms of monoclonal antibodies, these are plasma B cell hybridoma derived, which means that you take an immunized animals, you derive specific B cell clones from that immunized animals, you fuse them with myeloma cells, and then that gives rise to a hybridoma, which you can keep culturing to get your monoclonal antibody population over and over again. And then a recombinant is the most advanced technique where you perform in vitro cloning of your IgG sequences from the B cells of an immunized host. And once the best advantage about the recombinant technique is that once you derive your antibody sequences, there is no more involvement of animals in the whole process. So once you have the heavy and light chain sequences derived from B cells, you can keep cloning them over and over again to keep reproducing the same antibody over and over again. Now, in terms of the type, polyclonal antibodies are a heterogeneous population of antibodies which recognize multiple epitopes on the same antigen. Monoclonal antibodies are a homogeneous population of antibodies which all recognize the same epitope on the same antigen. Now, in case of recombinants, they are genetically defined. As I mentioned, that you actually derive the heavy and light chain sequences from B cells and then clone them and produce your antibodies. So typically they are monoclonals, but in the more recent times, we have started seeing a new class, and actually the multi-Rab recombinant antibodies that I'll be talking about at the end are multiclonal. Multiclonal recombinants are actually produced by mixing or developing a mixture of monoclonal or multiple monoclonal recombinant antibodies. Now, what are the advantages of these different classes? Polyclonals, because they recognize multiple epitopes, they offer high sensitivity. For monoclonals, sensitivity can be low sometimes, but they do have low background. They are much more specific, and they have minimal lot-to-lot variability. Now, recombinants, the process is very, very controlled. So they are highly specific. They have minimal background. They have high lot-to-lot consistency because you are pretty much using the same sequence over and over again to produce your antibodies. And you also have high data reproducibility because of the lot-to-lot consistency. Now, in terms of disadvantages, again, polyclonals, they can have lot-to-lot variability because they are animal derived. They can have higher background sometimes. And then in terms of monoclonals, although their specificity is much higher, they have lower sensitivity. Recombinant, the problem is that the main disadvantage is that it's a much more newer concept. So people are slowly trying to adopt it, but the selection is limited. So you may not find the species specificity or the host that you are looking for. So taking all these factors into consideration when it comes to secondary antibodies, polyclonal secondary antibodies are most widely used and are even highly recommended because they are the most highly sensitive. And indeed, most commercially available secondary antibodies that you might be using in your lab are already polyclonal. Now, monoclonal secondary antibodies are recommended for applications which require detection of specific primary antibody class or subclass. So one example of such an application would be, again, multiplexing between same host species primary antibodies, which have different subclasses. As I already mentioned in my last kind of topic, that Proteintech nano secondary reagents, they do allow, they are subclass specific, so they will allow you to multiplex different subclasses of, let's say, mouse IgG, and they are by nature recombinant monoclonal VHHs, or they are multiclonal. So sometimes they can be mixture of two monoclonal nanobodies or VHHs, and they bind again to primary antibodies in a species and subclass specific manner. So this is another example where we stained HeLa cells with two different antibodies which had the same host species, but they had different subclasses. So a mouse IgG2b antitubulin antibody and a mouse IgG1 CD147 antibody. And we detected them using monoclonal anti-mouse nano secondaries. So I just want to emphasize one more time that these nanosecondaries, they can be either single clones. So for example, the IgG 2B is, I think, a single clone, whereas the IgG1 is a mixture of two monoclonal antibodies. But that's the kind of the advantage of using monoclonal secondary sometimes. So if you're looking for a highly specific target, in this case, which is a mouse IgG subclass, a monoclonal secondary antibody might be the best choice for your application. Now, next, moving on to choice of conjugate. So the main consideration when it comes to the choice of conjugate is, of course, the application. So the conjugate is just a very, in simple words, the detection label that's attached to your secondary antibodies. And the choice of conjugate can depend on other factors other than application, such as the target or the properties of the conjugate itself. So this table kind of summarizes what are some of the secondary antibody conjugates that you might want to take into considerations depending on your application. For the purposes of this talk, we are going to focus on these two, immunohistochemistry and immunofluorescence. So when it comes to immunohistochemistry, you have quite a few options. You can definitely choose between enzyme-labeled secondaries if you are doing chromogenic IHC, and you have two options, HRP or poly-HRP, or you can use fluorescent labeled secondaries for fluorescent IHC. Sometimes you might want to actually enhance your signal or amplify your signal, and that's when you might want to use biotinylated secondaries. And I will be talking about that in a little bit more detail. And when it comes to of course, let's say IF applications, whether cells or tissues, you will have to use fluorescent labeled secondaries. But what type of conjugate or which conjugate, fluorescent conjugate you're using can be affected by several factors. So before moving into IF, I'll briefly talk about chromogenic IHC because it can sometimes be confusing whether you want to use HRP, poly-HRP, or a biotin labeled secondary. So how do you make that decision? So before moving into the details of that, let's take a quick look at the overview of the chromogenic IHC. So chromogenic IHC, the principle is that it utilizes chromogen and substrate combination for antigen detection. So basically, you start with your tissue preparation step, which I think I mentioned previously is pretty elaborate. So you have to fix your tissue samples, then you have to dehydrate them. Then there is your paraffin embedding, sectioning, and then again rehydration. So once you have done all of that, your sections are ready for antigen retrieval to expose any kind of masked epitopes. And then this is followed by quenching. Quenching is a step just which kind of blocks any endogenous peroxidases, which might sometimes can give non-specific staining. Then you do your usual blocking step to prevent any unspecific interactions with your primary antibody. Then you have your antibody incubations and amplification steps. So the antibody incubations and amplification step involves primary and secondary, and there can be more additional steps depending upon what level of amplification you want to achieve. And once you have done that, you want to do signal detection. So the most widely used combination of enzyme and substrate for chromogenic IHC workflows is HRP and DAB, where your secondary antibody is conjugated to the enzyme HRP, and DAB is a substrate which you add and it produces a brown color. So that's what gives you the brown color in your IHC samples. And then, of course, to provide a better contrast to your IHC images, you want to counter stain your samples with something like hematoxylin, which stains your nuclei blue, followed by mounting and imaging. So that's kind of the basic overview of the chromogenic IHC workflow. So the main step that we will emphasize on or talk about is the antibody incubations and amplification step, because it can significantly impact the quality of your IHC data. So when it comes to like the amplification step or the secondary antibody step, there are several options, right? Like, so I already talked about direct detection, where you can use a directly labeled primary antibody. You can use indirect detection, where you have your primary antibody unlabeled, but your secondary antibody is labeled with the enzyme, which in this case, let's say, is HRP. However, this indirect detection method, although it is more sensitive than direct detection, it may not be sensitive enough to amplify or give you the signal intensity that you're looking for certain low to moderate abundance targets. So that's when you can use these other techniques, which are called the avidin biotin complex detection technique, or ABC technique, or the labeled streptavidin biotin detection technique, or the LSAB technique, which actually utilizes the high affinity interaction between avidin and biotin for signal amplification. So they're a little bit different from each other. So just kind of briefly giving an overview for ABC detection, what you do is you have your primary antibody, then you have your biotinylated secondary antibody, and then you form separately, you form these large complexes between avidin and biotinylated HRP. Once you have these large complexes formed, you add it to your sample, which then comes and binds to your biotinylated secondary. And what you have in the process is like these really, really large complexes. And because avidin is tetravalent, so it can actually bind 4 biotin molecules, which in turn can bind HRP molecules. So that leads to signal enhancement. However, as you can see from the size, that this complex is pretty large, right? So that was kind of one major drawback of this method, that because of its larger size, it had difficulty penetrating tissues. And the LSAB detection method was actually developed as an improvement of this earlier method. So the LSAB detection method is pretty similar. You have your primary antibody, you have your biotinylated secondary antibody, but in this case, you used HRP conjugated streptavidin, which has a very similar structure to avidin, and you add it to your secondary antibody, which goes and binds to your biotinylated secondary. So in this case, you again have like you have multiple biotin labeled secondaries attached to your primary antibody, which allows you to amplify your signal, but the signal the complex size is much reduced, so you get much better tissue penetration and you also achieve higher sensitivity. But one major drawback of both these techniques is that it relies on the use of biotin, and that can cause background for a lot of IHC applications because there's some endogenous level of biotin in most cells, and some tissues have more biotin than others. When you do paraffin embedding or fixation, it helps you to get rid of that biotin to some extent. But again, when you do the antigen retrieval step, it can expose that biotin again. And especially when, if you're working with frozen tissue samples, biotin can be, the endogenous biotin can give you some really bad background. So to kind of address that issue came in this third method, which is a polymer-based method, where you had a polymer backbone, which carried several of the enzyme and secondary antibody molecules, and that allowed signal enhancement. The earlier polymers were, again, large structures which had trouble penetrating tissues, but nowadays we have these more advanced micropolymer or compact polymer-based secondaries, which allow efficient tissue penetration and also gives you high signal amplification. So that being said, basically what we recommend is that if you are doing IHC for the detection of moderate to low abundance target, we definitely recommend using poly-HRP conjugated secondaries. And to summarize, the reasons are that it results in significant increase in assay sensitivity compared to traditional HRP conjugated secondaries. Your poly-HRP conjugated secondaries will also give you much lower background compared to traditional avidin biotin-based amplification techniques like ABC and LSAB, which I just described. Also, the steps are much fewer. With poly-HRP, you simply have to use it as any other secondary antibody. You don't have those additional steps of, conjugating your avidin to your biotin-related HRP. So it is much more faster and requires fewer steps compared to traditional amplification methods like ABC and LSAB. And also it requires less primary antibody. So it helps you to conserve your primary antibody because it results in significant signal enhancement. So just to give you some examples of what I was just talking about. So here is an example of a data slide which shows comparison of TDP43 staining in mouse brain tissue sections. On the left, we have used our multi-rab HRP only conjugated goat anti-rabbit recombinant secondaries. And on the right, we have our multi-rab poly-HRP conjugated goat anti-rabbit secondary. And you can see that there's definitely a significant difference in signal intensity between the two images. Next, the other example I wanted to highlight here is that here we have done a side-by-side comparison between poly-HRP conjugated multi-rab secondary antibody. So that's the first image that you're looking at over here. But in the second and third images over here, we used a biotin conjugated secondary antibody followed by incubation with HRP conjugated streptavidin. So as I mentioned before, that use of biotin-related secondary antibodies can lead to higher background, and we clearly see that high background in this case. You can see that there is much higher background in this middle image over here, which utilized a biotin-related secondary antibody and HRP streptavidin. And this is furthermore proven by the third image over here, where it's a no primary antibody control. So even in the absence of primary antibody, we see this light background staining, which clearly says that there's some interaction between your biotin-related secondary and the endogenous biotin. Compared to these two, the first image, which used poly-HRP anti-rabbit, looks much more cleaner with much like lesser background. So with that, I'll move on to a little bit about IF workflows. So IF workflows, again, primarily the consideration is that which fluorescent conjugate you will use. Of course, it will depend upon your preference or how you want to build your multiplex panel, how many colors or targets you're looking at, but there are several factors which can actually influence staining intensity or brightness. So first of all, you have your that just the fact that different dyes have different properties, which in turn can impact brightness. So by properties, I mean like you can have your extinction coefficient, which means like how strongly a particular fluorescent molecule absorbs light at a particular wavelength, right? Or how quickly that absorbed light is basically converted into emitted light. So all those type of properties of a dye molecule can impact its brightness. Next is, what was the conjugation method that was used for conjugating that dye? This in turn can actually impact the degree of labeling, which is how many dye molecules are being carried by a particular secondary antibody. So that can also vary from one conjugate to another. Then third is compatibility of the dye with the imaging instrument. So you have to make sure that your imaging instrument can or has the appropriate filters and lasers for the dye that you're using. And then sometimes we have even seen that certain dyes, even with similar, emission excitation ranges from different manufacturers, can actually behave differently. So that being said, we do have some recommendations when you select your fluorophore. So we recommend that you perform side-by-side comparison of the same secondary antibody conjugated to various dyes for detecting multiple targets and sourced from the same supplier. So you want to make sure that a secondary antibody, when conjugated to multiple different dyes, performs consistently across multiple targets when you're sourcing them from the same supplier. Second is, we do need to take dye brightness into account when we are assigning fluorophores based on target abundance. So like, for example, as a rule of thumb, we recommend that if the dye is too bright or has a higher staining intensity, you would use them for the staining of low abundance target and vice versa. And then, of course, as I already mentioned, you have to always make sure that the excitation and emission maxima of the dye should be compatible with the lasers and detectors of the imaging instrument you're using, or with the other fluorophores if you're doing a multiplex panel. And lastly, the last suggestion we have is that fluorophores with red or far red emissions are much more recommended for samples with high autofluorescence. So for example, if you're doing tissue IF, it is recommended that you use the 647 conjugates, and you're expected to get much lower background than compared to if you are using a 488 conjugate. So here is an example of what I was just talking about, that staining intensity or brightness can vary from one fluorescent conjugate to another. In this case, what we did was we used the same multi-rab goat anti-mouse recombinant secondary antibodies, but they were conjugated to 4 different dyes, namely 4A coralite plus 488, 555, 594, 647, and we kind of tested it across 3 different targets: PRDX3, beta tubulin, TDP43C terminal, and we can clearly see that there is a difference in staining intensity between the 4 conjugates. And kind of looking at this data, for example, if you had to stain for beta tubulin, for example, you can see that the coralite plus 594 and 647, the signal looks pretty saturated, right? So in that case, you might want to use 488 or 555 for that target. So it is kind of important to do this side-by-side comparison of the same secondary antibody conjugated to various dyes. And then coming to the last factor for the selection of secondary antibodies are cross-adsorbed secondary antibodies. So before talking about cross-adsorbed secondary antibodies, I just want to quickly talk about like how polyclonal antibodies in general are produced and why would you want to cross-adsorb them, right? So generally speaking, polyclonal secondary antibodies involves immunization of an host animal, like for example, goat, and then you collect blood from that immunized animal, centrifuge it to separate out the serum. That's what actually contains your antibody. So you take your antibody from the serum and purify it, which yields your polyclonal antibody population. And you then want to validate it in relevant applications that you intend to use it for. And then you have your final antibody uh, population, right? So this is kind of the generic workflow of polyclonal antibody production. But it does have some caveats. So because of this production process, what we get at the end is an undefined pool of antibodies purified from the serum of an immunized animal. So the composition of antibodies in the polyclonal pool really is dependent on the natural immune response of the host, which can vary widely from one animal to another or when the same animal is immunized again. So that's something to keep in mind. So while this approach yields antibody population which is highly sensitive, the pain points associated with polyclonal antibodies is that they do tend to have high cross-reactivity with unintended IgGs, and they can have high lot-to-lot variability, which can lead to variability in experimental data. Now, the first point, high cross-reactivity with unintended IgGs can have a significant impact when you're using a polyclonal secondary antibody. So cross-reactivity from secondary antibodies can actually lead to high background when you are working with tissue samples, for example, because your secondary antibody can bind to endogenous immunoglobulins in the sample, which can give rise to high background. And the second limitation is that it can also lead to non-specific signal when the secondary antibody binds to off-target antibodies in multiplexing applications because of this high cross-reactivity. So to kind of address this issue, basically one solution is to use cross-adsorbed secondary antibodies. So cross-adsorbed secondary antibodies are traditional polyclonal secondary antibodies that have undergone additional purification steps to eliminate cross-reactivity against unintended IgGs from non-target species. And the way it is done is that basically your polyclonal antibody population is passed through a column which has these immobilized IgGs from other species or IgGs from other species or from other animal serum, and that kind of helps you to eliminate any kind of non-specific IgGs. And what you get in the flow through is just your specific IgG population. So, however, that being said, cross-adsorbed polyclonal secondaries, well, while they can overcome cross-reactivity issues, but they can still have some drawbacks. So they do tend to have decreased sensitivity because, as I said, that they are undergoing an additional filtration step, right? So you are losing some of those maybe sensitive species in your polyclonal population. So that leads to decreased sensitivity. And then not that they are always fully effective. Earlier, I had shown you a data image of pre-adsorbed rabbit secondary antibodies against mouse, which were pre-adsorbed for mouse IgGs, but it still showed residual cross-reactivity with mouse IgG subclasses. So it's not fully effective, and the specificity can still vary from one lot to another, right? Because at the end of the day, they are still polyclonal. And these cross-adsorbed polyclonal secondaries also tend to be very, very expensive. Sometimes they are even double the cost of your regular non-cross-adsorbed secondaries. So while cross-adsorbed polyclonal secondaries do offer a solution for applications like IHC or IF, where you are having issues with background and non-specific signal, they do also pose some challenges. And that sort of brings us to the last topic for today. So to address all of those pain points, what we did was we developed our multi-RAB recombinant secondary antibodies, which can really address all of those challenges. And they kind of bring together all the benefits of polyclonal and monoclonals together and help you get the best quality images. So just to kind of give you an overview of how we produce them, how we develop them, and what are the advantages that they offer. So our multi-Rab recombinant secondary antibodies are mixtures of recombinant goat monoclonal antibodies that recognize multiple complementary epitopes on the same IgG. And this mixture, each recombinant clone in this final antibody mixture, we individually characterize them to ensure the highest level of performance. So we make sure that every clone that goes into the final mixture has been individually characterized. So if you look at the image on the right here, the workflow diagram, it kind of gives you a overview of the development process. So what we did was we started with hundreds of recombinant secondary antibody clones. Then we did an initial screening with Western blot and ELISA for preliminary selection of clones. Now the antibodies which passed the selection process, they were then characterized for their binding sites. So the binding sites for each recombinant clone was individually characterized. Then we individually conjugated each clone. Each conjugated clone was then tested for cross-reactivity against various species, for high background for low background and high brightness. And then we picked the best clones, obviously, and developed multiple mixtures in different combinations of the top performing clones, which also offered optimal epitope coverage, right? So as I mentioned earlier, that we did characterize the binding sites for each of those clones. So we wanted to make sure that all the clones that go into that mixture um, uh, cover enough epitopes or complementary epitopes to give you the optimal detection levels. And then each of those mixtures were functionally characterized using various applications. And ultimately, we selected the antibody mixture with optimal performance, which then goes into final production and QC. So as you can see that the whole process has been pretty rigorous. So it goes through each of those clones, goes through several screening and validation steps just to ensure that the final mixture you have gives you the minimal background, the brightest signal, and the best performance across multiple applications. So just to summarize the features and the benefits and the pain points that the multi-Rabs address. So multi-Rabs are multiclonal mixtures of recombinant monoclonal antibodies. So they do combine the high sensitivity of polyclonals with superior specificity of monoclonals. The pain point addressed here is that traditional polyclonal secondaries are highly sensitive, but they can have high cross-reactivity with unintended IgGs leading to non-specific staining. Second, the multi-Rab secondaries are recombinantly produced, which means that they are not purified from animal serum. They have high lot-to-lot consistency, which leads to reproducible experimental results. And it also has security of supply, right? So which means that we can keep producing the same antibody again and again because they are sequence defined. Traditional polyclonal secondaries, on the other hand, can have high lot-to-lot variability, which can lead to variability in experimental results. Also, our multi-Rab secondaries are stringently validated, as I just described. The benefit is that they have minimal cross-reactivity with off-target species and optimal performance in all applications. And the pain point addressed is that traditional polyclonal secondaries do tend to have high cross-reactivity with off-target IgGs, making them unsuitable for certain specific applications, like, let's say, multiplexing. So the data image over here just gives you kind of an overview of how the cross-reactivity levels of the multi-Rabs look like. So what we did here, we compared the cross-reactivity of our goat anti-rabbit recombinant secondary antibody with non-cross-adsorbed and cross-adsorbed secondaries from two leading competitors. And so basically, and we used direct ELISA for this method, and you can clearly see that. So the first blue bar over here represents our multi-Rab goat anti-rabbit secondary, and then the second and third bar, the orange and yellow, those are non-cross-adsorbed and cross-adsorbed secondaries from competitor one, and the fourth and fifth bars are non-cross-adsorbed and cross-adsorbed secondaries from competitor two. And you can clearly see over here that the cross-reactivity levels of our multi-rab goat and anti-rabbit secondary was pretty comparable or even sometimes better than the cross-reactivity levels of cross-adsorbed secondaries from the two competitors. So I just want to also emphasize that what we have to offer for our multi-rab portfolio at the moment. At the moment, we have like two species specificities, anti-mouse, anti-rabbit, and they were all raised in goat or derived from goat. And you can see that the conjugate options available for each one of them are 7. So HRP, then we have 5 correlate conjugates, and also polymer HRP for chromogenic IHC applications. So since we are talking about IHC and IF today, so most of the correlate plus dyes that we have should be sufficient for most of your IF workflows. So we have 488, 555, 594, and 647. And your poly HRP is the one that I spoke about earlier, offers advantages of higher sensitivity for in chromogenic IHC workflows. This is just some illustration of data on the top row over here. It just shows that so all of the images over here kind of looks at dual labeling with one mouse and one rabbit antibody. So in the top row, both secondaries used were our multi-rab recombinant secondaries, and on the bottom row here, basically we paired our multi-rab secondary with a traditional polyclonal secondary. That is to say that they are compatible. So you can still use the multi-rab secondaries with traditional polyclonal secondaries within a multiplexing application if you wanted to. And then I just also want to emphasize, because this is a question that we get from our customers quite often, whether they work on both frozen and FFPE samples or not. So they do. So we have validated all our correlate plus conjugated multi-rab secondaries in both FFPE and frozen sections. So you can use them for both tissue types. And as I mentioned, I also wanted to share some data for our poly HRP conjugated multi-rab. It is very suitable for the detection of low abundance targets. So if you're working with any low abundance target and having trouble detecting them in your chromogenic IHC workflows, definitely consider trying out our poly HRP conjugated multi-rab secondaries. And the other thing I want to mention here, that our poly HRP conjugated multi-rab secondaries come in a ready-to-use formulation, so you don't have to dilute it any further. You can directly apply it as it is. And last but not the least, another question that we often comes across is that whether they work for both manual and automated staining or not, because a lot of labs or IHC service providers, they do rely on automation quite a bit for IHC workflows. So yes, our multi-rab poly HRP secondaries have been validated for both manual and automated staining. So that being said, just to conclude, I just wanted to summarize that our secondary antibody portfolio at Proteintech really consists of 3 different product lines. So we have our multi-rab recombinant secondary antibodies, traditional polyclonal secondary antibodies, and nano secondary reagents. So once again, the multi-rab recombinant secondary antibodies, these are recombinant goat multi-clonal secondary antibodies. We have just 2 species reactivities at the moment, anti-rabbit, anti-mouse, and both of them are available in 7 conjugates, correlate plus 488, 555, 594, 647, 750, HRP, and poly HRP. So if your application involves these 2 species reactivities and these conjugates, we definitely highly recommend you to try these multi-rab secondaries out. You will see that you're able to get really the best quality image that's there. You will get high sensitivity, minimal background all at once. And today's webinar actually will also allow you to get a free sample of this product if you have not tried it out already. But that being said, as you can see that the species reactivities for multi-Rabs at the moment are limited. So to kind of compensate for that, we still offer traditional polyclonal secondaries, which are polyclonal secondaries purified from animal serum. So for this, we have 10 species reactivities. We have 15 plus conjugates, including a lot of the traditional dyes like FITC, PE, and we also have some fragment antibodies available like Fab2. So if your applications require any of these needs, then please feel free to try these out. So we have all of these in stock as well. And then last but not the least, the nanosecondary reagents, which are recombinant alpaca VHHs conjugated to fluorescent dye. As I mentioned earlier, that they are subclass specific and therefore they're suitable for same species multiplexing. So if you want to have a secondary antibody which allows you to multiplex, let's say, between different mouse IgG subclasses, this will be your choice of secondary antibody. And then, of course, if you're working with super resolution imaging, this is also what you want to use. So I think with that, I'm at the end of my presentation. And thanks for your attention. And Abel and I can, Frida, we can all take questions if there are any left. Yeah.
All right, great. Thank you so much, Swati. This was such a great topic, and you did a really great job kind of giving a comprehensive overview of, the importance of selecting your secondary antibody. A lot of times when we're doing staining, we put a lot of time and effort into just the primary antibody selection. And so I really hope that everyone kind of the takeaway from this today is that there should be a lot of considerations going into your secondary antibody as well. So, really big thank you, Swati, for the very comprehensive overview, and Abel for going through and answering. I think you answered pretty much all the questions in there. So I hope everyone's questions got answered. If they haven't, there is a slide at the very end that will have Swati's email, so you can absolutely reach out to her at any time if you have any questions. I do want to just do a couple questions just live. I know we're a little over, but I just think these are some good questions that came in. So the first question that I just kind of wanted to address live is, can someone use a secondary against IgG heavy and light chains for an IgG2A primary antibody?
Yeah, so this is a very good question. So I believe Swati mentioned in her slides, mentioned that most of the commercial IgG with AH plus L can recognize all isotype classes. So IgG2A without exception, so they can also bind with IgG2A like other IgGs. However, we have used a lot of other competitor secondaries in our test. We see that they have bias in some cases, like you get secondary from vendor A, they will react with the IgG1 more than IgG2A, while you use the others, you have the opposite results. So, but in most cases, they should be strong enough for signal development.
Great. Thanks so much, Abel. The next question that I just wanted to address real quick is, someone had asked, is it bad practice to use antibodies raised in a given species for samples originating from that species? So for example, using an antibody raised in mouse for mouse slices.
Yeah, we got a couple of questions asking about mouse on mouse samples. So in our experiences, we see some customers have issues in these cases. We analyze their causes of this background. It's always that they see the background because of there's residual blood in the samples, because there's the endogenous IgG on the samples. So when you apply the secondary to the samples, they can detect the endogenous IgG. So what you have to do is to get rid of those IgG, which was by removing of the blood completely. However, it is very difficult to remove all of the blood in the animal. So before you sacrifice the animal, it's better to run a perfusion to wash away all of the blood in their vessels. So that would help in most cases. So what if you go back to 3 slides, we have a lot of co-staining on the IF slides. Go back, please, on the back. So there's one slide that you have a lot of immunofluorescence pictures on the back. Yeah, this one. So take this one, for example. Let's look at the first one. So it's mouse cereblin, and we also have a mouse monoclonal antibodies, which is combining D28K. So the red one, right? So you don't see any non-specific signal or any background in the red signal, right? So that is because we removed the blood completely when we prepare the samples. So if you can do that, so you won't see too much issues on the background. However, for some blood-rich tissues like spleen and kidney, it is very difficult to remove the blood completely. Then you can use some blockers if necessary, like there are some commercial blockers or some non-conjugated IgG secondaries. So you can also use those as blockers to block the endogenous IgG, so that can prevent the secondaries to bind them. So it is possible to overcome that issue. But if you can choose primary from another host, then that would be make life easier.
Great. Thanks so much, Abel. There's 2 more questions that I wanted to just address. The first one is, at Proteintech, we put a lot of emphasis in our validation data, and most of our validation data is done in FFP sections. Do the multi-Rabs work in frozen sections?
Yeah, it does. So Swati has 1 or 2 slides showing the data.
So this is the slide which I just wanted to again kind of point out that yes, all our Coralite plus conjugated multi-Rab secondaries have been validated in both FFPE and frozen sections.
Awesome, thank you. And then the last question is kind of a culmination of some of the questions that we got. There were people who were asking about whether we had secondaries for different species. So I guess the overall question is, in terms of the pipeline, are there any new species that you guys are planning to kind of focus on for secondary antibodies, or different new fluorophores or anything like that? Is there anything that our audience can expect in the future, in the near future?
For species, we don't have any on our pipeline yet, but we are working on expanding the applications. So like we are working on biotin conjugation, and at the same time we are also looking for new dyes, and especially someone asked, do we have any dye beyond the higher further wavelength than 647 and lower than 488. So those are our goals at this moment. So we will make more directions using current raw material of unconjugated secondaries. So make them to have more application for the detection.
I've noticed there's definitely been someone who's been really cheering for axolotl. So seems like it's a very hard find for secondary antibodies. So I got you. I will whisper it to our R&D team. So if you could go to kind of towards the end, Swati, I just have a couple slides to kind of just end our webinar for today. So I just wanted to end by mentioning that, in addition to being an antibody supplier, we also really value providing researchers with beneficial resources to help with their research. So we offer resources to help with both IHC and IF workflows, including in-depth guides, technical videos, detailed answers to FAQs, and blogs that are showcasing, tips and tricks for immunostaining in different tissues and different conditions. So you can.
So after
Yes.
Sorry to interrupt. There's one more question which is very interesting. I want to highlight it here.
Sure. Yeah.
So yeah, the question is, I suppose I have the rabbit primaries for protein A and B. Can I use the flexible systems to have correlate 488 conjugated to rabbit anti-A and use multi-rab secondaries 47 for protein B? So with a co-staining method for indirect and direct. So the answer is yes, it's doable. We do this now and then, but you have to make sure that there's an order. So you have to do the indirect first. Otherwise, your secondary recognize both of the primaries. So you have to add anti-B and then apply the secondaries of multi-rab. Then you put the correlate flexible conjugated protein A antibody. Then you are ready to stain the slides. So it is doable. You just have to follow the orders. So indirect first and then direct after that.
Great, thanks, Abel. Yeah, that's actually a really great question. We get asked that kind of frequently about, the protocol of how to use flexible and kind of like a traditional primary secondary. So yeah, no, thanks for catching that and for answering that question. So just to kind of get back to this, so if you guys are interested in any other resources that we do have for IHC and IF, feel free to scan the QR codes and it will take you to our website and you can kind of access all that information there. And so to the next slide, this is the last slide that we have. I just wanted to mention that as an attendee of today's webinar, you will be receiving a follow-up email that will include a recording of the webinar. We will also include a PDF of the slides. We'll also have a certificate of attendance that will be mailed to you separately, and then a link to request a free multi-rab secondary sample. So if you scan the QR code, you can request your sample right now, or you can wait until you get the follow-up email and we'll have the link there as well. So we do just ask to please wait at least 1 week for the follow-up email to be sent. It takes a little bit while for everything to kind of get in order and to get sent out. So just allow us 1 week for that follow-up email. So with that, I just want to thank everyone again for attending and for all the great questions that we received. And Swati for her presentation, Abel for answering all the questions. I don't know, Swati, if you have one more slide at the end that has your email on it. So if you do have any other questions for Swati or Abel, feel free to email Swati and she can forward them over to Abel at swati@ptglab.com. If you have any questions about the event in general, please email us at events@ptglab.com. And if you liked today's event and want to learn about any of our upcoming events, please do follow our social media pages because we post about all our upcoming events on there. So I hope everyone has a great rest of your morning, afternoon, or evening, depending on where you're tuning in from. And I hope to see you guys all at future events. Take care.